Very warm welcome to Kalang platform. Igniting Curious Mind has on date the 8th June 2026. Chapter 16 Excretory Product and their Eliminations. Animal accumulates ammonia, urea, uric acids, carbon dioxide, water, ions like Na plus, K plus, Cl minus, phosphate, sulfate, etc., either by metabolic activity or by any other means like XXI ingestions. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of eliminations of these substances with a special emphasis on a common nitrogenous wastes. Ammonia, urea, uric acids are the major form of nitrogenous wastes. Excretion by animals. Ammonia is the most toxic form, requires large amounts of water for its eliminations. Urea, whereas uric acids being the least toxic and can be removed with a minimum loss of water. The process of excretion ammonia is aminotelism. Many bony fishes, aquatic and amphibians, aquatic insects are aminotelic in nature. Ammonia has its readily soluble in general excretion by diffusion across the body surface or through the gill surface, as in fishes, ammonium ion. Kidney do not play a significant role in its removal. The terrestrial adaptations of necessitates the productions of lesser toxic nitrogenous waste like urea, uric acids for conservation of water. Mammals, many terrestrial amphibians and marine fishes mainly excretes urea is called as ureotelic animals. Ammonia produces metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidney. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain the desired osmolality. Reptiles, birds, land snails, insects excrete nitrogenous waste has uric acids in the form of pellets and paste with a minimum loss of water and are called uricotelic animals. The survey of animal kingdom presents variety of excretory products structures. In most of the invertebrate, these structures are simple tubular form, whereas the vertebrates have a complex tubular called organ called kidney. Some of these structures are mentioned here. The photonephroid or the flame cells are excreted product in platyhelminthes, flatworms, or planaria. Example planaria. The rotifers, some annelids and cephalochordata, amphioxus, and the protonephridia are the primarily concerned with ionic and the fluid volumes regulations, osmoregulations. The nephridia are the tubular excretory structures of the earthworm and the other annelida. Nephridia helps in removal of nitrogenous waste and maintain the fluid ionic balance. Malpighian tubules are excreted structures of most of the insects, including cockroaches. The Malpighian tubules helps in removal of nitrogenous waste and osmoregulations. The antennal glands and the green glands perform excretory functions in the crustaceans like prawns. Human excretory system. In humans, excretory system consists of a pair of kidney, one pair of uterus, a urinary bladder, and urethra. Kidney are reddish brown bean-shaped structures situated between the levels last thoracic, third lumbar vertebral cross close to the dorsal inner walls of the abdominal cavity. Each kidney of an adult human measures 10 to 12 centimeter length and 5 to 7 centimeter width and 2 to 3 centimeter thickness, with the average weight of 1 to 120 to 170 grams. Towards the center of inner concave surface of the kidney is a notch called as hilum, through which the ureter, blood vessels, and nerve enters. The inner to the hilum is a broad funnel-shaped space called as a renal pelvis, which with the projections called as a callus. The outer layers of the kidney is tough capsules. Inside the kidney, there is a two zones, that is outer cortex and inner medulla. The medulla is divided into few colonial colonial masses, medullary pyramids projecting into these calyces, calyx, and the cortex extends between the medullary pyramids as a renal columns called as columns of Bertins. Each kidney has a nearly 1 million of complex tubular structure called nephrons, which are functional units. Each nephron has two parts, the glomerulus and the renal tubules. The glomerulus is a tuft of capillaries formed by the efferent arterioles, a fine branch of renal arteries. The blood from the glomerular is carried away by the efferent arterioles. The renal tubular begins with a double-walled cup-like structure called Bowman's capsules, which encloses the glomerulus. The glomerulus, along with the Bowman's capsule, is called as a Malpighian's body or the renal corpuscles. The tubular continues further to form a highly coiled network proximal convoluted tubules. The hairpin structure, that is Henle's loop, is the next part of the tubules, which has a descending and ascending limbs. The ascending limbs continues A hairpin-shaped Henle's loop is the next part of the tubules, which has a descending and ascending limbs. The ascending limbs continues to another highly coiled tubular regions called as distal convoluted tubules. The distal convoluted tubules of the nephrons opens into a straight tubes called as collecting duct, which converge opens into renal pelvis through a medullary pyramid and is in the callus. The Malpighian corpus and PCT and DTC of the nephrons are situated in a cortical region of the kidney, whereas the loops of Henle's dips into the medulla. In majority of nephron, the loops of Henle is too short and extend very little into the medulla. Such nephrons are called as cortical nephrons, and some of the nephrons, the loops of Henle's is very long and runs deep into medulla. These nephrons are called juxtamedullary nephrons. The efferent arterioles emerges from the glomerulus, forms a fine capillary networks around the renal tubules called as pertubular capillaries. A minute vessels of this network runs parallel to Henle's loop, forming U-shaped vasa recta. Vasa recta is absent and highly reduced in the cortical nephron. Urine formation. Urine formation involves three main processes: glomerular filtration, reabsorption, secretions takes place in different parts of the nephron. The first step in urine formation is filtration of blood, which is carried out by glomerulus and its glomerular filtrations. On average of 1100 to 1200 ml of blood is filtered by the kidney per minute, which constitutes roughly 1/5 of the blood pumped out of each ventricles of the heart in a minute. The glomerular capillaries blood pressure causes the filtration of the blood through three layers: the epithelium of glomerular blood vessels, the epithelium of Bowman's capsules, and the basement membrane between these two layers. The epithelial cells of Bowman's capsules are called as podocytes and are arranged in an intricate manner so as to leave some minute space called as filtration slits or slit spores. The blood is filtered so finely through these membrae that almost all the constituent of the plasma except the protein pass on to the lumens of the Bowman's capsule. Therefore, it's considered as a process of ultrafiltrations. Amount of filtrate formed by the kidney per minute is called as glomerular filtration rates. GFR is healthy individuals is approximately 125 ml per minute, 180 liter per day. The kidney have built-in mechanism for regulations of glomerular filtrations. One such efficient mechanism is carried out by juxtaglomerular apparatus. JGA is a special sensitive region formed by a cellular modification in the distal convoluted tubules and the efferent arteriole at the locations of their contact. At a fall in GFR can activates the JG cells to release a renin, which can stimulate the glomerular blood flows and thereby the GFR back into normal. The comparisons or the volumes of filtrate formed per day, 180 liter per day, that of the urine released 1.5 liters, suggests that nearly 99% of the filtrate has been reabsorbed by the renal tubules. This process is called as reabsorptions. The tubular epithelial cell is in different segments of nephron perform this either by active or passive mechanism. For example, Substances like glucose, amino acids, Na plus, etc. in the filtrate are reabsorbed by actively, whereas nitrogenous waste are absorbed by the passive transport. Reabsorption of water also occurs passively in initial segments of the nephron. During urine formation, the tubular cells secrete substances like H plus, K plus, ammonia into filtrate. Tubular secretion is also important steps in urine formation as it helps in maintenance of ionic and acidic balance of the body fluid. Functions of tubules. The proximal convoluted tubule is lined by a simple cuboidal brush border epithelium which increases surface area of the reabsorption. Nearby, nearly all the essential nutrients, the 70 to 80% of the electrolytes and water are reabsorbed by the segments. PCT is also helps to maintain the pH and ionic balance of the blood fluid by a sensitive selectively secretions of hydrogen ammonia into filtrate by absorption of HCO3 from it. Henle's loop reabsorption is a minimum in its ascending limbs. However, this region plays a significant role in maintenance of a high osmolarity by a medullary intestine fluid. The descending limbs of loops of a Henle's is permeable to water but also impermeable to electrolyte. This concentration of filtrate is moved down and the ascending limbs is impermeable to water but allow transportation of electrolyte actively or passively. However, the concentration of filtrate passed down and it is diluted due to the passage of electrolyte to a medullary fluids. The distal convoluted tubule, the conditional reabsorption in Na plus 1 water takes place in the segments. The DCT is capable of reabsorption HCO3 and the selective secretions of hydrogen, potassium ions and NH3 to maintain the pH and sodium potassium balance in the blood. The collecting duct. The long duct extended from the cortex of kidney to the inner parts of medulla. The larger amount of water could be reabsorbed from this region to produce a concentrated urine. The segments allows passages of small amounts of urea into medullary intestinum to keep up the osmolarity. It plays a role in the maintenance of pH of ionic balance of the blood by a selective reabsorption as H plus K plus ion. The mechanism of concentrations of the filtrate. The mammals have the ability to produce a concentrated urine. The Henle's loop and vasa recta plays a significant role in this. The flow of filtrate in the low two limbs of Henle's loop is opposite direction and thus forms a countercurrent. The flow of blood through two limbs of vasa recta is also countercurrent pattern. The proximity between the Henle's loop and vasa recta as well as the countercurrent between them help in maintain the increasingly osmolarity towards inner medullary intestine. The 300 ml of osmolarity per osmolarity in the cortex to that of 1200 ml of osmolarity in the inner medulla. This gradient is mainly caused by NaCl in the urea. The NaCl is transported in ascending limbs of Henle's loop, which is exchanged with the descending limbs of vasa recta. NaCl is returned to the intestinum by ascending portions of vasa recta. Similarly, small amounts of urea enter the thin segments of ascending limbs of Henle's loop, which is transported back into intestinum by collecting tubules. The above described transports of substances facilitated by special arrangements of Henle's loop and vasa recta is called as countercurrent mechanism. The mechanism helps to maintain a concentrations gradient. In the medullary intestinum, the presence of such intestinal gradients helps in an easy passage of water from the collecting tubules, thereby concentrating the filtrate urine. The human kidney can produce nearly 4 times concentration than the initial filtration formed. The regulations of kidney functions. The function of a kidney is efficient, monitored and regulated by a hormonal feedback mechanism involving a hypothalamus, JGA, and to a certain extent the heart. The osmoreceptor in the body are activated by changes in the blood volumes, the body fluid volume, ionic concentrations. An excessive loss of fluid from the body can active their receptors and stimulate the hypothalamus to release the antidiuretic hormones or a vasopressin, from which the neurohypophysis ADH facilitates the water reabsorption from later parts of the tubules, thereby preventing the diuresis. An increase in the blood fluid volumes can switch off the osmoreceptors and suppress the ADH release to complete the feedback. ADH can also affect the kidney function by its constrictory effect in the blood vessels. This causes an increase in the blood pressure, and an increase in blood pressure can increase the glomerular blood flow, and thereby the JJ plays a complex regulated role. A fall in the glomerular blood flow or glomerular blood pressure, the GFR can activate the JG cells to release the renin, which converts angiotensinogens in the blood and angiotensins 1 and further angiotensins 2. The angiotensin 2 begins a powerful vasoconstrictory, increases the glomerular blood pressure and thereby the GFR. Angiotensin 2 also activates the adrenal cortex to release the aldosterone. The aldosterone causes the reabsorption of Na plus and water from the distal parts of the tubules. This also leads to an increase in the blood pressure and GFR. This complex mechanism is generally known as renin-angiotensin mechanism. An increase in blood flow to an atria of the blood and can cause a release of atrial natriuretic factor. ANF can cause us a vasodilation, that is dilation of blood vessels, and thereby decreases the blood pressure. ANF mechanisms therefore acts as a check of the renin-angiotensin mechanisms. Micturations. The urine formation of nephron is ultimately carried to urinary blood, where it stores till the voluntary signal is given by a central nervous system. The signal is initiated by the stretching of urinary bladder as it gets filled with the urine. In response, the stress receptors of the walls of the bladder sends the signals to CNS. The CNS passes on the motor messages to initiate the contractions of a smooth muscles of the bladder, and simultaneously relaxation of urethral sphincter causes the release of the urine. The process of release of a urine is called as a micturition, and the neural mechanism causing it is called as a micturition reflexes. An adult human excretes an average of 1 to 1.5 liters of urine per day. Urine formed in a liter yellow-colored watery fluid which is slightly acidic, pH 6 point, has a characteristic odor. On an average of 25 to 30 grams of urea is excreted per day. The various conditions can affect the characteristics of urine. Analysis of urine helps in clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. The presence of a glucose, that is glucosurine, and the ketone body ketonuria in the urines are indicative of diabetes mellitus. The role of organs in excretions. Other than the kidney, the lungs, livers can also help in elimination of excretory waste. Our lungs removes large amounts of CO2, approximately 200 ml per minute, and more significant quantity of water per day. Our liver is the largest gland in our body, secretes bile-containing substances like the birulinum and the biliverdinum and cholesterol, degraded steroid hormones, vitamins, and drugs. Most of these substances ultimately passes out along with the digestive wastes. The sweat and the sebaceous glands in the skin can eliminate certain substances through their secretions. Sweats produced glands, and the sweat gland is a watery fluid containing the NaCl and small amounts of urea, lactic acids, etc. Though the primary function is the sweat is to facilitate a cooling effect of body surface. It also helps in removal of some of the waste mentioned above. The sebaceous glands eliminate certain substances like sterol, hydrocarbons, waxes to the sebum, and this secretions provides a protective oil covering for the skin. Do you know a small amount of nitrogenous wastes could be eliminated through the saliva too? The disorders of excretory product. The malfunctions of a kidney can lead to accumulation of urea in the blood, a condition called uremia, which is highly harmful and may lead to kidney failure. In such patients, urea can be removed by the process called as hemodialysis. During the process of hemodialysis, the blood drained from the convenient artery to be pumped into dialysis units called artificial kidney. The blood drained from the convenient artery is pumped into dialysis unit after adding an coagulants like heparin. The units contains coiled cellophane tubules surrounded by a fluid, dialysis fluid, having the same composition of that of a plasma except the nitrogenous waste. The porous cephalophone membrane of the tubules allow passage of molecules based on concentration gradient. As nitrogen waste are absent in the dialysis fluid, the substance freely move and thereby clearing the blood. The cleared blood is then pumped back into the body through the vein and after adding antiherpin to it. This method is boon and for thousands of uric patients all over the world. Kidney transplantation is ultimately method to concentrations of renal failure, kidney failure. The functioning kidney is used to transplantations from a donor, preferably close relative, to minimize its changes of rejection by the immune system of the host. The modern chemical procedures have increased the success rates of the complicated technique. The renal calculi, the stored or insoluble mass of crystallized salts, oxalates, etc., formed within the kidney. The glomerular nephrites, the inflammations of a glomeruli kidney. Summary: the main nitrogenous substance contains substance iron, carbon dioxide, water, and accumulated in the blood body have to be eliminated. The nature of nitrogenous waste formed from their excretion vary among animals, mainly depending on the habitat, availability of the water, ammonia, urea, uric acid, and major nitrogenous waste excreted. The photonephridia, nephridia, Malpighian tubules, green glands, and the kidneys are the common excretory organs in animals. Not only they eliminate nitrogenous waste, but also help in eliminate a maintenance of ionic and acidic balance of the body fluid. In humans, excretory system consists of one pair of kidney, a pair of ureter, a urinary bladder, urethra. Each kidney has millions of tubular structures called nephrons. Nephron is a functional unit of kidney and two portions of the glomerular and renal tubules. The glomerular is a tuft of capillaries formed from afferent arterioles and the fine branches of renal arteries. The renal tubular starts with a double-walled Bowman's capsule. It's further differentiated into proximal convoluted tubules and Henle's loops and distal convoluted tubules. The DCTs, many of the nephron joins common collecting duct, many of which ultimately opens into a renal pelvis through which a medullary pyramid. The Bowman's capsule encloses the glomerular to form a Malpighian or the renal capsules. Urine formation involves three main processes: filtrations, reabsorption, secretions. The filtration of non-selective process performed by glomerulus and the glomerular capillaries. Blood pressure: about 1200 ml of blood is filtered by the glomerulus per minute to form 125 ml of filter of Bowman's capsule per minute. The JGA is a specialized portion of the nephron, plays a significant role in regulation of GRFNL. 99% of reabsorption of filtrate takes place through the different parts of the nephron. The PCT is a major site of reabsorption and selective secretion. HL primarily helps in maintaining the osmolar gradient and the within the kidney intestinum. The DCT and the collecting duct allow the extensive reabsorption of water, certain electrolytes, which helps in osmoregulation. The H plus K plus NH3 could be secreted by filtration by the tubules to maintain the ionic balance and pH of the body fluid. The countercurrent mechanism operates between the two limbs of the loops of the Henle's and those of the vasa recta capillaries parallel to the Henle's loop. The filtrate gets concentrated as it moves down the descending limbs but also diluted ascending limbs. The electrolytes and the urea are retained in the intestinum by the arrangement. The DCT and the collecting duct concentrate the filtrate about 4 times more from 300 osmolarity to 1200 osmolarity per osmolarity, and a excellent mechanism of conservation of water. Urine is stored in a urinary bladder till the volumetry signals of CNS carries out. It's released through the urethra. The micturition, the skin, lungs, liver also assist in the excretions process. Thank you for watching. You're tuned into current platform Igniting the Curious Mind. Thank you.